Hello students, welcome back. Today I am Radha from Ardent Progressive School, going to start the same chapter that is we were discussing in the last class, photosynthesis in higher plants, right? So in this chapter we were discussing about the photosynthesis, and we have done that there are two main reactions, okay, or we can say two main steps. What are they? Light reaction and dark reaction. Right, and we have done that in light reaction, what is happening in the light reaction, there will be the photosystem 1 and photosystem 2. If I will talk about the sequence wise, then the first there is a photosystem 2 and the photosystem 1, and all the light reaction will take place where? In the chloroplast. And in this chloroplast, what is that specific site? If it is a chloroplast in any of the cell, okay, if it is a chloroplast, and you will see the cross section, you can see that inside this chloroplast, what is there? There are thylakoids. Okay, what are there? There are thylakoids. They are arranged like this, stack like, okay, and these thylakoids are interconnected with what? With the lamella, stromal lamella. We have done this thing. So if I will talk about the specific site of the light reaction, it is in the membrane of this thylakoid. Okay. Now if I will take one thylakoid from this granum, the whole unit is known as what? The granum. So I am not talking about the whole granum. I have taken one of this one. If it is one of the thylakoid, which I am showing here. Okay. I have taken this thylakoid and have drawn this structure like this. Is it correct? Okay. So here what is going to happen in this membrane? Okay, in this membrane you can see that inside there is a lumen. We have done this thing that inside this there is a lumen, and the outside there is a matrix. Okay, the matrix which is present in the chloroplast, it is known as what? It is known as stroma. Okay, it is what? It is the stroma. So what happens here again? The outer word. Okay, the outside you can see there is a stroma, right? So what is going to happen here? For example, this is the whole chloroplast, right? I am drawing with this structure. Now what happens during this time? That in this membrane of what? Membrane of thylakoid. So in the membrane of thylakoid there will be light reaction with the help of what? With the help of photosystem and photosystem 1, photosystem 2 and the photosystem 1. As the light will come from here, okay, so accordingly with the help of wavelength, we have done P700 and P680 nanometer. Okay, so according to the wavelength, what happens? The electron will get excited in this membrane only. Okay, due to this, what happens? There will be removal of proton. We have done with the help of the chemiosmotic hypothesis. Right. So what I want to mention it here, that we have done all the steps which takes place inside the light reaction. Okay. And if I will talk about the light reaction, what can be the product? Okay. There will be formation of energy like ATP and NADPH. Right. So this will go where? In the stroma. Where it is going? It is going into the stroma. So the another reaction or the another step which we have done, that is a dark reaction. Today we are going to discuss about the whole detail of the dark reaction. Light reaction takes place where? In this membrane of the thylakoids, and after forming these things, it will send its byproduct towards the stroma, okay, where it is going to send its byproduct in this stroma. So this stroma is the site of what? Is the site of the dark reaction. Okay, so we have to continue from here that how the dark reaction will take place in this particular stroma. Now we have done this thing also, that in light reaction what happens, that in light reaction there must be a presence of light. It means that it is a light dependent reaction. Okay, but if I will talk about the dark reaction, why we have said that it is a dark reaction? It doesn't relate with any of the dark condition. It doesn't mean that it is happening in the dark condition or at night. Both the light reaction and the dark reaction takes place in the presence of light only. Okay, if there will be no light, there will be no light reaction, and ultimately there will be no dark reaction. So these are correlated with each other. So here we can say that as the light reaction will complete in a sequence way, there will be a dark reaction in this stroma, and in this dark reaction it is not dependent on the light, whether it is dependent on the product of the light reaction. Okay, that's why we said it is a dark reaction, and this dark reaction is also known as the biosynthetic pathway. Why? In this particular stroma, what we are going to do in this, we will study that there will be a formation of the food, because ultimately, if I will talk about the whole reaction of the photosynthesis, then if you will recall the reaction, what happens during the photosynthesis? In the presence of light, there must be CO2 and water. And another thing, what we are going to study, we are going to study that these are the inorganic compounds. Okay, from these inorganic compounds, we are making the organic compound, that is glucose. So ultimate aim of this photosynthesis is what? The formation of glucose. Glucose is what? It is a bioproduct, right? So here what we are going to do, we will study that how this phase act, because it is a biosynthetic related to the living and synthesis of the glucose molecule. Okay, so up to here it is incomplete. Light reaction has completed, but now the time is required for forming the food. You have excited the electron, okay, and you got the byproduct, but if there will be no dark reaction, so there will be no formation of food here. Okay, so there must be thing that there should be what? There should be the formation of food. So how it will take place? So on this basis, we have discussed that in this cycle or in this phase, what is going to happen? There will be assimilation of CO2. The first thing, that ATP and NADPH, it is coming from this thylakoid membrane and from the outside. And from the outside, what is coming here inside it? The CO2. Okay, and CO2 is very, very important for the formation of glucose. Okay, the CO2 is the carbon which is coming from the CO2. It is the only thing which is responsible for the synthesis of the glucose molecules. Okay, now CO2 will come from outside and the energy will come from this thylakoid membrane, and here it will get react. Okay, it will get react, and due to this reaction, what will happen? There will be formation of food. So how this will take place in the plants? So on the basis of this assimilation of CO2 here, we have categorized the plants into two types. What are they? C3 type and the C4 type. Okay, what we are saying? C3 plant and C4 plant. And what is the difference between these two types of plant? That they are following the C3 pathway and it is following the C4 pathway, right? Any of the plant, all these chloroplasts are present where? In the mesophyll cells, right? So in the mesophyll cell, there is a chloroplast, and inside the chloroplast there is a thylakoid membrane as well as the stroma. So here we have to discuss all about it. Okay, now comes to the C3 pathway. What do you understand by the C3? Because most of the plant, or we can say most of the green, whether it is a C3 plant or the C4 plant, okay, the pathway C3 will be common for the both. But in case of C4 plant, there is one difference. Okay, along with the mesophyll cell, there is some different anatomy which we are saying Kranz anatomy. Okay, what we said? Kranz anatomy, and they are having the bundle sheath cells also. So along with the mesophyll cells in the C4 pathway, there is a one more type of cell, that is the bundle sheath cells. Okay, so today we are not going to discuss about the C4 pathway. So first we will discuss about the C3 pathway. So C3 pathway takes place in all the photosynthetic plants, right? And why it is different from the C4 pathway? Because we know that CO2 is coming from outside. Okay, and during that time when the temperature of the environment will get high, okay, we know this thing that if temperature will get rise in the atmosphere, what happens in the plant? The plants is trying to close its stomata. So during the extreme temperature, the stomata will get closed. If stomata will get closed, the CO2 will not be able to enter inside the cell. In which cell? In the mesophyll cells, right? So this is the thing about the C4 pathway. Because in case of C4 pathway or the C4 type plant, they are having some different anatomy which will prevent this thing. Okay, and if if this thing is happening during the daytime, what happens? If the stomata of the plant, if the stomata of the leaves will get closed during the daytime, what will happen? We know that the CO2 will not be able to enter inside it, and along with this there will be more production of oxygen, and we know that there is a splitting of water molecule in this thylakoid membrane. So what exactly it is happening in the thylakoid membrane? There will be splitting of water. This splitting of water is releasing the oxygen towards the outer side, right? And there will be formation of ATP and NADPH. So this oxygen will go outside through the stomata if the temperature will be normal at the outside. Okay, if the temperature of outside will get rise up, what will happen? The oxygen will get accumulated here. What will happen? The oxygen will get accumulated in this stroma, and it will not be able to go outside. And oxygen will not be able to go outside, and CO2 will not be able to enter inside. So ultimately what will happen? This oxygen will takes place at cycle inside the stroma, and this oxygen is not of use for the formation of glucose, because for the formation of glucose what we require the carbon dioxide, which is going to come from the outside, which has been stopped in this condition. Okay, so this condition is known as what? This condition is known as photorespiration. Okay, what does it mean? That photo means in presence of light, respiration means that there will be breakdown of food or there will be release of oxygen. Okay, during this respiration, what will happen? That we are releasing the CO2, and plants also release the CO2. Okay, so in this condition oxygen will get fixed here, and it will not be able to form any food, and ultimately it is going to release what? It is going to release the CO2. Okay, so this is the condition which we have to discuss. That what is photorespiration? Photorespiration is that which the respiration which takes place in presence of light. Respiration is good. Okay, respiration is good during the night hours. That's why we know that the night hours there will be respiration of plants, and we should not sleep under the tree. Why? Because during the night hours the plants used to respire. Okay, but in the daytime, what is the work during the daytime? In the daytime, the work is that there should be a entry of CO2. Okay, if there will be entry of more CO2, then there will be formation of more food. Okay, so in this cycle, we are going to study that how CO2 will get fixed here in the stroma. What are the conditions? So for that one, we have to study the C3 cycle, and this C3 cycle, it is also known as the Kelvin cycle. Why? Because it is given by the Kelvin, right? So let's start with the Kelvin cycle. If I will talk about the cycle, it is divided into 3 steps. What are they? 3 steps. The first step is carboxylation, second step is reduction, and third step is regeneration. Okay, so we have to understand that what exactly it is happening in each and every step. So let's start with the first one. This, what I have written here, ribulose 1,5-bis phosphate. What it is? In short, we can say it is RUBP. What it is? RUBP. So if I will talk about in the stroma, the CO2 which is coming from the outside through the stomata, it will fix with the RUBP which is present here. Okay, so RUBP is the primary acceptor of CO2, or we can say the acceptor of CO2 is what? RUBP. If RUBP will not be there, then no one is going to accept this CO2, right? So this is very, very important, this RUBP. Now, when it will enter inside this one and it will attach with the CO2. So I have written here that CO2 which is coming from the outside as well as water. Okay, as well as the water, it will combine with RUBP. Okay, when it will combine with RUBP, what it is going to give? It is going to give 3-phosphoglycerate. 3-phosphoglycerate is nothing, it is a glyceric acid. The thing which I have mentioned here, the ATE. Okay, when we are talking about the carboxylic acid, we are adding this word ATE. Okay, so we can say it is what? It is a 3-phosphoglyceric acid, right? Now you can see that RuBP is a 5 carbon compound. Okay, it is 5 carbon compound. So if I will draw 1, 2, 3, 4 and 5. Okay, so this RuBP is 5 carbon compound, and when it will add with the CO2, so this CO2 will give one more carbon, right? When it will give one more carbon, what it will become? It will become 1, 2, 3, 4, 5, and addition to it, this carbon is coming from the CO2. Now it will become what? It will become 6 carbon compound. Okay, so this reaction is taking place somehow in between these steps, which is not being shown in your NCERT also, but the reaction is what? After adding with CO2, what it will form? It will form 6 carbon compound. Now this 6 carbon compound is unstable. So what it is going to do? It will split this 6 carbon compound. It will form the 3 carbon compound. Okay. So what we will get here? We will get 2 molecules of 3 carbon compound. Right? How? We can say that it will form 3 carbon here, 3 carbon here. Right? So what is going to happen? We are going to get 2 molecules of what? This 6 carbon compound, which is formed by the addition of RuBP and CO2. Right? Along with this, one more thing, the enzyme which is going to involve here. What is that enzyme? The enzyme name is Rubisco. Okay? Rubisco. What do you understand by Rubisco? It means that RuB, ribulose by bisphosphate, as well as this C and O is relating to the carboxylase. I am writing here carboxylase and oxygenase. Carboxylase and oxygenase, and both are what? These are the enzymes, because there is ASE. So now what is the role of this enzyme Rubisco? It means that this ribulose bisphosphate carboxylase oxidase enzyme have the tendency to fix both carbon as well as oxygen. Okay, so that's why this enzyme helpful here to form this 3-phosphoglycerate. Right? So we can say that the RuBP will combine with CO2 in presence of this Rubisco enzyme, and it will form the 3-phosphoglyceric acid, and there will be 2 molecules. Why? Because it is the 3 carbon compound. What it is? It is a 3 carbon compound, and earlier it was the 6 carbon compound. Now the question arises that what do you understand by this Rubisco? I said this Rubisco will fix both the carbon as well as oxygen. And what is our requirement here in the C3 cycle? That it should fix the carbon dioxide. Okay, why? Because it is having the more affinity to fix with what? With fix the CO2. But it doesn't mean that it will not fix with the oxygen. That's why I said during the photorespiration. During the photorespiration, what will happen? The CO2 will not get enter inside this cell. Due to this, there will be an accumulation of oxygen, and during this condition, during the condition of photorespiration, this Rubisco enzyme, instead of fixing this carbon dioxide, it will fix what? It will fix the oxygen. Okay, so that's why there is a Rubisco enzyme, because if there will be a photorespiration in the photosynthetic plant, then this Rubisco having the ability to fix this oxygen also. Right? So here we have done that Rubisco enzyme is very important to fix this carbon dioxide which is coming from the outside. Now, the first step has been completed, that is carboxylation. What do you understand by carboxylation? It means that when there is a fixing of carbon dioxide, it is known as the carboxylation. If I will talk this reverse about the carboxylation, it is decarboxylation. What do you understand by decarboxylation? When there will be removal of CO2, okay, and where it will take place? During the respiration, because we know that during the respiration there will be release of CO2. So in that condition we will study about the decarboxylation. But here what is carboxylation? It is a fixation of CO2 in this cycle, okay, with the help of this RuBP and enzyme Rubisco enzyme ribulose bisphosphate carboxylase oxygenase. Right? Now comes to the another step, that is the reduction step. Why we said reduction? Because there is a removal of hydrogen molecules. Okay. Now along with this, when we will get 2 molecules of 3 carbon compound, and this 3 carbon compound is 3-phosphoglyceric acid, it means that at the third position, Okay, at the third position there will be a addition of some phosphorus molecule. Right, now comes to this step. In this step, what is required? We require the energy, and from where this energy is going to come? From the membrane of the thylakoid, the byproduct of the light reaction. Okay, so during the light reaction, what happens again? There will be removal of oxygen outside, removal of ATP and NADPH outside in the stroma. So what will happen in this reaction? The energy ATP and NADPH which is coming from the thylakoid membrane after the light reaction, it will get attached with this 3-carbon compound, that is 3-phosphoglycerine. Okay, so all this thing is happening in this stroma. Right. So with the help of this ATP and NADPH, what is going to happen? It will add the phosphorus molecule, because what is the role of this ATP? ATP is providing the phosphorus. Okay, and after providing this phosphorus, it will convert into the ADP. ADP is what? Adenosine triphosphate. It means that this triphosphate will give one phosphate molecule and it will form what? It will form the ADP. So what is coming outside? Inorganic phosphate. Along with this, the NADPH which is forming here, it will also provide it here and it will release in form of NADP positive. And during this time what will happen? This acid, we know that oxidation is what? Oxidation is alcohol to aldehyde, aldehyde to acid. This is the chain in chemistry. But if I will talk here, just reverse of it. Reduction means just reverse of oxidation. So from this acid, what we are going to make here? Aldehyde. Okay, just opposite than alcohol. So here what we will get? Again we will get the 3-carbon compound, and along with this 3-carbon compound we will get this food. So this formation of food takes place here where? In this Calvin cycle. That's why we said in the stroma the dark reaction will take place. This dark reaction will form the glucose inside this stroma. Okay, and this is the ultimate thing of the photosynthesis. So after that we will get the sucrose and starch, because these are things which is present in the plant in form of storage. The glucose is stored in case of plants in form of starch. So here, from here we will get the ultimate result, that is the food. Right, and after getting the food, it should again go to the formation of REP, and this cycle should continue. Okay, if this cycle will not get continued, what will happen? We will not get this food, because again it has to make the RUBP. It means that again it has to make 5-carbon compound, and we know that here we are having triose phosphate, and it means that 3-carbon compound and 3-carbon compound like this, and we are having 2 molecules. So here again we will make only 1 molecule. Okay, again we are making 1 molecule. 3 and 4, 5 we will take from here, and it will send back to the RUBP. And when we are forming the same thing, we are regenerating it. What we are doing? We are regenerating, and the third step which will come here, the regeneration. And this regeneration again needs some energy, and this energy in form of ATP. So when we are providing this ATP, it will regenerate the RUBP and releasing the ADP here, right? So the whole step having 3 things. What are they? Carboxylation, then reduction, then regeneration. Right, and in this thing what we have done, that we are in starting we are having the 5-carbon compound. Addition to it we are getting 6-carbon compound, then it will get splitted into the 2 molecules of 3-carbon compound. So what we are saying, up to here, from this part to this part, in this carboxylation and reduction, we are having 2 molecules. Okay, what we are having? 2 molecules. And here we are having again the 1 molecule. So what we can say, how many ATP and how many NADPH we are using in this whole C3 cycle. So from here we can say that there is use of 1 ATP here and 1 NADPH. Okay, if there will be 2 molecules, for 1 molecule we said we are using 1 ATP and 1 NADPH, but for the 2 molecules we are having 2 ATP, okay, 2 ATP and 2 NADPH, right? And here 1. The total we are having how many ATP? 3 ATP. 2 ATP from here and 1 ATP from here. So we can say that 3 ATP we are using in this 1 cycle. Okay. Now how many NADPH we have used? We have used the 2 NADPH. Why? in one cycle or for one molecule 1 NADPH is there, for 2 molecules there will be 2 NADPH, right? So these are the energy which we have used here. But if you will recall the step of the photosynthesis, what we know about it, we are saying that there will be addition of 6 carbon dioxide. Why? Because if I will talk about only one CO2, so we will not get the glucose, because glucose is what? C6H12O6, and we have done that this 6 carbon is coming from the CO2, right? So if this 6 carbon is coming from the CO2, so there must be a 6 carbon dioxide, right? So what is going to happen? There will be 6 CO2 will get enter inside it, okay? But that's why we are saying this cycle will continue for the 6th time. Okay, so this for in one cycle when we are having one CO2, so like this it will continue for the 6th cycle. So in one cycle we have used 3 ATP. Okay, in one cycle we have used 3 ATP, but the complete cycle will be what? If there will be 6 round. Okay, if there will be 6 round, so how many ATP is required? 18 ATP. We can say for the synthesis of one glucose molecule. For the synthesis of one molecule of glucose, what is required? We require 18 ATP, and how many NADPH? We can multiply it. 2 6 are 12 NADPH. Okay, so you can see that how many CO2 is going to enter inside the cycle? 6 CO2 in, right? And here if we will multiply it, 18 ATP and here 12 NADPH. Okay, so after the cycle what we will get ultimate product, that is our glucose, and ultimately it will get continued in this cycle. Okay, so today we have completed about the C3 cycle, which takes place both in the C3 plant as well as C4 plant. Okay, and in the C3 cycle we are fixing the CO2 with the help of which enzyme? Rubisco enzyme. Okay, so for today that much is there. Thank you.